Dear friends, welcome to Biology Made Easy. Today we'll discuss about the Hershey and Chase experiment. In the previous video, we have discussed the Griffith experiment where it was proved that DNA is the genetic material. But there also I have told you that scientists at that time didn't believe it. They thought that it is protein. But in 1952, Alfred Hershey and Martha Chase, they proved that the genetic material is actually DNA. It is not protein. So how they proved it? They took one culture medium in which there is E. coli, and to this they also gave one bacteriophage. Now bacteriophage, phage means eater. That means bacterial eater, means bacteriophage means a virus. And the structure of bacteriophage is such that when it will infect, it behaves like a syringe. When injection is given to somebody, the needle remains outside, but when the syringe is pushed, the medicine goes inside. So similarly, when the bacteriophage will infect the bacteria, the genetic material will go inside, but the protein coat will remain outside. So in one medium, they put heavy isotope of S35. This is radioactive. And when the virus will grow within it, the new viruses which will come out, they will have S35 in their protein code, because sulfur is a constituent of amino acid like methionine and cysteine. So naturally, the S35 will be present in the protein code. In another set of experiment, they perform the same experiment, but here they put P32. Phosphorus is a component of nucleotides and ultimately nucleic acid, and it is not a component of sulfur. So whenever P32 is given, the nucleic acid will have radioactive phosphorus. So in this way, they isolated two separate bacteriophages, T2 bacteriophage. Now in one case, there is S35 present in the protein code. In the other case, P32 that is present in the genetic material. Now when both were allowed to infect E. coli, first point, infection, just as I have told you in the case of syringe, they will now infect. Now the genetic material will enter into the E. coli while the protein coats will remain outside. By means of blending means grinding. Now the protein coats which were present in the outer side, now they are separated, and the genetic material will now replicate inside. In the third set, that is centrifugation. This is important. First point, infection, then blending, then centrifugation. In centrifugation, when the E. coli was centrifuged, the virus particles were separated from the bacteria, and when they analyzed where the radioactive substances are present, it was found in the first case where S35 was used, it is present in the protein coat present outside in bacteriophage. It is not present in the E. coli. Similarly, in the second case where P32 was used, that was found within the virus, the newly formed virus, but the protein coat which is present outside, it has no radioactive substance. That means it is the DNA which acts as genetic material, not the protein. So this experiment of Hershey and Chase conclusively, unequivocally proved that DNA is the genetic material. Here sometimes students confuse whether the question is asked, who proved that DNA is genetic material? Sometimes they tell that it is Hershey and Chase, but it is wrong. Very MacLeod and McCarthy proved that DNA is genetic material, but Hershey and Chase proved unequivocally, conclusively, that DNA is genetic material. So, depending upon the question, the answer will be different. So, once DNA the genetic material is proved, now the second question came. What is that question? There are some organisms where there is no DNA, only RNA is genetic material. So in that case, which will act as genetic material? This experiment was conducted by Frankel Conrad, taking two types of viruses. One is tobacco mosaic virus, the other is Holmes ribgrass virus. In case of TMB, when they were separated, the protein code and RNA. Similarly, in case of Holmes ribgrass virus, there is protein and RNA. When the RNA of TMB and protein of Holmes ribgrass virus were joined together, now this virus will behave like whom? Like TMB, because the RNA belongs from TMB. Now if the RNA of Holmes ribgrass virus and protein will be joined together, now this will behave like Holmes ribgrass virus because the RNA belongs to this. So from this, Frankel Conrad proved that RNA is genetic material, but here you have to mark one thing. RNA is genetic material when DNA is absent. When both DNA and RNA are present, the DNA is genetic material. So now we have proved that both DNA and RNA can act as genetic material, depending upon the situation. So what should be the important characters of genetic material? That is again very important. So there are few points. What are the characteristics of genetic material? Number one is a replication. Because when the cell divides into 2, 4 and likewise, all the cells are equal. So unless the genetic material has capacity to duplicate, capacity to replicate, then it is not possible. All cells cannot be equal. So that is number one point. Second point, they should be structurally very stable. There should not be any fluctuation in their structure. Third point, they should have a capacity of slow changes, which we can tell in a single word, mutation. Unless there is slow changes in the nucleotide sequence, all the organisms will look like same. And we are observing diverse type of organisms, even in the same organism, different types of phenotypes are there. It is because of the slow mutation. The fourth point, the genetic material must follow Mendelian inheritance. So these are the 4 points or characters which a genetic material should possess. So I hope you have understood it. Now we'll discuss few questions. Number one question. Who proved that RNA is genetic material? A choice, Griffith. B choice, Avery et al. Et al means co-workers, Avery, MacLeod, McCarthy. C choice, Hershey and Chase. D choice, Conrad. Frankel Conrad. Now come to the second question. Which radioactive substances were used by Hershey and Chase? A choice, AS35, P35. P choice. S35, P32, P35. Here S35, P32, here S35, P32. C choice. S32, P32. D choice. S32 and P35. Now come to the third question. Which phage was used by Hershey and Chase for their experiments? A choice, lambda phage. B choice, T2 phage. C choice, T4 phage. D choice, T5 phage. So after going through the video, you will be able to answer all these questions. This is very important from examination point of view. So I hope you have understood it perfectly, and hope we'll meet you again.